The other genetic thing is how quickly you metabolize different drugs.
Yep.
And so some groups are taking the spectrum of all the drugs and metabolism and telling you which medications you take in a normal dose or where you have to reduce your dose.
Yeah. So dosing is an important one. And that's, I think, probably one of the areas where 23andMe got in trouble, because they were giving you your information about warfarin and 3 or 4 other drugs. They're the exception, okay? And even in those cases, it's not 100%. Okay? So most drug responses are actually very complex, just like what we're talking about. There's hundreds of genes involved, and we can't predict what they are. There are exceptions, like warfarin will potentially be useful, but I've talked to, so like epilepsy, for example. We know that what drugs are used. We know that there are variants that affect that. But it turns out translating that to the clinic to affect dosing is not necessarily as useful as actually just the tampering that goes on anyway, because everybody's different, okay? So for a lot of that genetic dosing to be really effective, it has to be spot on. And there's so much among individual variability that it's not clear yet whether that will be that useful, okay? So yes, pharmacogenetics is a huge open field, but my impression is that in fact the hype on that is greater than anywhere else. That the actual research behind pharmacogenetics has got a long way to go. Okay, but it will come and it will revolutionize treatment in probably more like a 10-year framework than a 2-year framework. That's my view.
Right.
Yeah, I'm sorry, thank you. Yeah.
Are you aware of any current successes with pharmacogenomics and adolescent onset psychiatric disorders? What's the current thinking on it?
The current thinking on adolescent onset psychiatric disorders. So there's a lot of work going on autism and schizophrenia and ADHD and so forth. Turns out that all of those diseases have a shared genetic etiology to some extent. So there's the same genetic risk factors. In the field of human genetics in research, there are 2 basic models for genetics. One is called the rare variant model. And it says that every case of schizophrenia or autism is different, has a different genetic etiology. The other one is the common variant, common disease, or infinitesimal model, and that says that there are thousands of genes contributing, and if you're above a threshold, then you have disease. And the fusion of them is to say that the second one is correct, but it takes an extra insult, whether it's the environment or whether it's a mutation to get you over there. And that's certainly the way I view it. So what's happened is that you've had sequencing of thousands of cases in the last few months, actually, or the last 6 months. And what you see is an enrichment for genes involved in certain neuronal pathways. Synaptogenesis, for example, in autism is enriched. But it's not at the point where you can say, this is the cause of this particular child. Now, there are some cases. There are some things called copy number variants that are definitely segregating with autism. There are a few genes which are significantly mutated in autism, but they each explain collectively only maybe 5% of the cases of autism. The rest is this very complex risk that I'll sort of explain in the next slide. Okay? So there was a feeling that the psychiatric disease and metabolic disease were different. But as we learn more and more, we actually see that they probably have a lot in common. It's just different types of genes in each case. Okay. So actually, before I get to that, so why aren't we all rushing out and getting our personalized genome sequence? Well, one is cost, okay? So for about $500, you can get the light version, or if you were prepared to pay $99 to heavily subsidize 23andMe, you could get that. $3,000 today for the whole genome sequence. That's just to get the data. To interpret the data is add another $20,000, okay? But Illumina, as I said, is now doing it for about $1,000. There are companies out there who claim that they'll be able to do it for $100. In fact, you'll be able to get a thing that looks just like, a thumb drive that looks just like this, plug it into your laptop, put a dollop of, well, I said saliva, but maybe a droplet of blood, something in there, and it'll give you your sequence overnight. That's a dream. There's a company that sort of claims they'll be able to do that. And, well, they've been claiming they can do it for the last 5 years. But the theory is beautiful, so they will be able to do that at some point. So cost is one. Privacy is another one. I mean, you cannot guarantee privacy of this data. There's no way that Julian Assange could come along and just dump everything from the NIH database on the world at some point if you wanted to. So I can't guarantee that privacy, but it's obviously a very big deal for medical companies to try to protect patient rights. Your genomic data is something that most of us hold very, very privately, okay? And in fact, participation in any genetic study, this is one of the problems we have, is that we kind of guarantee that privacy. But it turns out that there are ways to identify people in studies, from surnames, matching Y chromosomes, things like that. Okay, so there are certainly concerns there. Insurance is another one. So people tend to be somewhat scared about what their knowledge will mean, not just to them, but to their insurers. So we have in America now the Genetic Non-Discrimination Act, which means that your health insurers and your employers cannot use your genetic information to discriminate against you, okay? But it's fuzzy and gray when it comes to life insurance. It's fuzzy and gray when it comes to if you know your data and you don't report it to your health insurance company. I had an exam question. No, none of them laughed at it. I thought it was a funny question. I said, what happened if an airline decided to discriminate against your obesity genotype? They could recoup a lot of money by charging people for their obesity alleles, okay? Well, that's not going to happen. But people don't really know what, I mean, I think most of us aren't really sure. We sort of know that there's a Genetic Non-Discrimination Act, but we're really nervous about what it means in reality, and those rules can always change. Then there's this basic anxiety, okay? So you've got this ratio of, am I going to learn that I'm going to get Alzheimer's disease when I'm 55? I don't want that knowledge compared to what I might get that is useful, okay? So we don't know enough about biology as lay people to be able to really make those decisions about whether the cost-benefit of participating in this is worth it. Now, there are studies starting to come out that sort of say that when you sit down and you educate people, then a lot of those anxieties go away, because sort of people see that there's not many cases where you're going to learn something really terrible. Okay? In fact, what tends to happen is that there are what we call false positives. So someone will get told that they have a particular mutation, and it's known that, you know, somebody with this mutation has some condition like, what's the peripheral neuropathy? Well, say Tay-Sachs disease or something. So you have a mutation, it predicts that, but you don't have that. Well, there's two reasons for that. One is because the databases of these thousands of genes may be wrong. Okay, so there's a lot of false information out there. And the other one is that there's incomplete, what we call penetrance. Okay, so before I became a human geneticist, I was a fly geneticist. I used to do things like making flies grow legs on their heads, kind of for fun. But actually, we were learning a whole lot. At least the NIH thought it was worth funding. But what happened is you could take, that's a pretty dramatic thing, right, to have legs on your head, okay? But it turns out you can take that mutation and cross it into different genetic backgrounds, and the legs will completely disappear in some backgrounds, okay? Same with an oncogene. The equivalent in a fly of an oncogene makes the eye of the fly really rough. You can take that mutation and cross it into different backgrounds, and it will completely go away. Okay? That's because our genomes are robust. That's my major research interest, is how genomes have evolved, it's called canalization, to really suppress the effects of all of these mutations. There's so much mutation happening all of the time that if we didn't have these robust systems, we'd all be dead many, many times over. So our genomes are very well able to absorb and deal with this variability. The book that Sherry mentioned, It Takes a Genome, my argument in that book is that we've got this disequilibrium between the environment in which humans evolved 100,000 years ago and our current environment where we all go to McDonald's every day and sit in front of the TV for 4 hours. That disequilibrium is exposing this genetic variation. Our systems can't deal with that insult to it as much, okay? I'm digressing. So, but it all comes down to, as well, this consent issue. I mean, how can you consent to getting your own genome sequenced if you don't really understand it? And if the geneticists don't really understand it as well, okay? But in fact, I would say we do that all the time anyway. I mean, how many of you go to a doctor and the doctor says, well, your platelet levels are high and you've got a high level of this or that, and you don't really know what that is. And yet you say, yeah, doc, okay, I'll take this medication. So it's not a whole lot different in this case. The difference is, and if you saw Ann Wozniacki, the founder of 23andMe, speak, she said the reason that she set that company up is because she wants to change the culture. She wants us to be invested in our own health. And that starts with self-knowledge. If we know more about ourselves, we're more likely to make smart decisions. We have control instead of our doctor having control. So this is my good colleague Atul Booth at Stanford's idea. So his personalysis is going to do this. So basically the idea of this is to say, well, we can take the prevalence of every one of these complex diseases, which currently for diabetes in America is about 25%. Well, it's not that high yet. Will be next year, 25%. Okay, so it's about 15%. And you can take a person's genome and you can say, well, you are in this extra risk category. Okay? I'm going to take each of your variants that I know influence diabetes risk, and there's 30 of them that are known, but we know that there are probably 500 more that we don't know yet. And I'm just going to multiply my baseline risk by the effect of each one. So this person actually have 5 bad genotypes, so they're in a bad state. But most of us will have this one will go up and that'll go down, and it'll sort of zigzag around, and we'll be somewhere in the middle for risk. But the idea is that you can do this for each one of these 20 or so chronic diseases we care about, and you can rank yourself relative to everybody else as to your likelihood of developing that disease, and then you multiply it by the baseline risk. Okay? So if I've got, 10 times as much risk of ulcerative colitis as everybody else, it doesn't really matter. My risk is still less than 1% because it's a very rare disease. It matters to that person, but it doesn't matter to most of us. But if you do the same thing for depression or obesity, then it does matter because the levels of those are so very high. Okay? And then what they want to do is to sort of put that all into a pattern where they say, well, here's your risks for coronary artery disease and obesity and diabetes and osteoarthritis. Here's what you're doing. Okay, so you smoke and you don't exercise and you don't go to yoga. All right? If you did, this bit here is going to shrink. Okay? So that's what we want, is a visualization that says that here's my genotype, my genome, here's my behavior. I want to be able to integrate those, incorporate them, and get to a point where I say, well, look, here's me on my current behavior, here's me if I change my lifestyle. That will be the dream. Yeah.
And when you looked at Ken Brigham's group,
yeah.
Did you look at that? So here's your risk of this, it's high. If you do this, this, and this, it'll decrease?
So great question. So intriguingly, so we couldn't give genetics back to people. Well, I'm still seeking IRB approval to do that. So the part of the consent for that study was we'll take the genetics, but we won't give it back to you. And because it's sort of the IRB sort of felt that it's just too, we don't know how to deal with that yet. So we're working through that. So if we do mock data, people want it. So I think people want it. Most people want it. Okay. But what we can do is say, well, each person, we can go to their records and say, what did you care about? Was it your weight? Was it your sleep patterns? Was it your bone density? And it turns out, at least for the metabolic stuff, it made no difference. The people who said they were cared about their weight lost weight about the same rate as everybody else. But where it did make a difference, and the numbers are pretty small, maybe I shouldn't say this, but it looked like for psychological conditions it made more of a difference. I mean, mental health is the starting point for physical health, is what Ken would say. And people who focused on mental health do seem like to do a lot better. Okay? We know very little about the genetics of mental health, so that's not good news. Okay? But this is kind of how it works, okay? So here's, so you've got 20 genes that we know influence height. In fact, it's now up to 500 genes, get that. They've looked at 500,000 people by this genome-wide association studies, not because they're interested in height, but because everybody who studies in any study has their height taken, okay? So they can do this. This was 3 years ago. And even though these 20 alleles only explain about 1% of height, in other words, if you took away these 20 genes, you wouldn't notice a difference walking around the street. But nevertheless, the people who have 30 tall alleles, so in other words, so for every gene you've got 2 what are called alleles, one from mum, one from dad. So if you get the one that increases height from mum and the one that increases height from dad, you've got 2. If you get the decreasing, you've got 0. Okay? And now you just add them up over all 20 genes or all 200 genes, and you can get your risk score, okay? So it turns out that people out here are on average 3 centimeters taller than people down here, okay? So there's a 6 centimeter difference between the tails. Now, as we get that up to more like 100 genes, then that difference becomes more like 10 or 12 centimeters, okay? And for height, it's trivial. Okay, no one really cares about. Well, maybe parents do, because height is the best predictor of income in life. Okay, so parents actually, that's probably one thing that people would really want. That's why I need a pay raise, Sherry, because I'm short. I've got to make up for my genes. Okay. Okay, But, you know, Danny DeVito could easily be out here. All these other genes are short ones. He just had tall ones here. And you know, Yao Ming could be down here, okay? So the error bars on this are massive, all right? Nevertheless, you can do that. So let's imagine now doing this for all of these common diseases. So we can all get a readout of where we are, okay, with respect to cardiovascular disease and so forth. Okay, and so that's what we've done. That's what we're thinking about, is generating something. Now, I'm not the data visualization guy. Alex is, so he'll take over sort of from here. But this is my conception. So I said, okay, let's do this. And we've got something like 10 different domains of disease that we care about. So there's going around them, immunological diseases, so inflammatory bowel disease, or just something as simple as how quickly we recover from the flu. You've got respiratory disease, so COPD or asthma. You've got cardiovascular disease, so atherosclerosis or maybe cardiomyopathy or maybe arrhythmias. You've got musculoskeletal disorders, so arthritis and muscle wasting and so forth. You've got metabolic disease, particularly diabetes, but dyslipidemias and so forth. You've got cognitive disorders, so by that I mean Alzheimer's and Parkinson's and early onset cognitive decline. You've got psychiatric diseases, under which I include depression, which is sort of the World Health Organization regards as the next diabetes of the 22nd century. But that would also include schizophrenia and ADHD and so forth. Clearly, you've got oncology is a major concern to most of us, okay? So we can formulate these risk factors in each of those domains. And now the question is, how do you integrate that with your clinical information that you're getting from your doctor anyway? And so we want to think about it in certain ways, okay? So here's somebody. This is just one person's profile. It's actually a hypothetical person, okay? So they have major depression. So, well, how this radar plot works is that each axis goes from low genetic risk in the middle to high genetic risk on the outside, okay? So I'm not calling it prediction. I'm calling it classification. So it says where you are relative to everybody else, all right? And then the dot says something about your clinical risk. So that's what we've been working with from your visits to the doctor, and maybe it incorporates family history as well, because we know that family history is important. In fact, 95% of Americans agree that having your family history of disease is important. 95% of Americans don't agree on anything, but they agree on that. But 30% of Americans actually have their family history, even a weak one, and only 10% of Americans' family physician has their family history. Okay? So we talk about genomic medicine, but there's a whole lot of just real simple stuff we can do before we get there. Okay? Now, so this person has high risk genetically of depression, and it turns out if we measure their Beck Depression Index, we measure their social function score, their isolation, and we measure their mental health score, they're really bad for those as well. So this is concordant. Their genetics is bad, and their social isolation is bad, and they're clinically depressed. I don't know, so what can we do with that? But that's a perfect storm. And maybe somebody who was down here for depression and had all these other things, maybe that would be motivating to be different, okay? So that's the example here. So hypertension. This person has extremely low genetic risk for hypertension. And yet, she's at the top end, the top percentile for systolic blood pressure, for arterial stiffness, and for Framingham risk factor. In fact, this is somebody in our cohort who came in exactly like that, top 1%. She didn't know her genetics. I know her genetics, okay? I don't know her, but I know her genetics. Her participation in the cohort, she came back down to normal. Okay, that would be the sort of success story we'd be looking. So you'd say, okay, my genome tells me I'm not at risk. My clinical profile says I'm really in a bad state. Maybe it's something I'm doing. I've got to cut the salt. I've got to stress less, okay? And then alternatively, you can have a situation where you have maybe a very high genetic risk for diabetes, and yet all of your clinical variables are absolutely fine right now. So this is the example that happened to another Stanford professor, Mike Snyder, a few years ago, who decided to take his blood every week for 2 years and do every omic profiling he could think of. And it looks like, I'm going to skip over one slide because it's not important, it looks like this, okay? Horrible data visualization, and it probably cost $200,000 of his startup. But it got him a Cell paper, and that's what matters to academics, okay? And he got 2 viral infections during the first 18 months. And and his genome sequence told him he's at high risk of diabetes, type 2 diabetes, even though there was none of it in his family, okay? After the second viral infection, his blood glucose levels started going whoop, and he actually became pre-diabetic. And he changed his behavior and he came back to normal. Now, he also measured everything else. He did his transcriptome, his microRNAome, his proteome, his metabolome, his antibodyome, his bloodome, his everything elseome, and it costs a lot of money. And we don't all need that. But there are aspects of that, all of those other things there that potentially will be useful and will become cheaper, and we'll integrate that. So he calls this an IPOP, an integrated personal omics profile. That's potentially the next wave of genomics. It doesn't just take your genome, but takes the readout of your genome, which is the step between your genome and your health. And that's sort of a 15, 20-year framework as to where that's going to come in. And it'll only happen if we can visualize that information better. So the question is whether cancer treatment currently is using more information than just the genome sequence, if it's using information on expression of genes. And the answer is not as much as you might think. So there were companies that came out a few years ago. I know a company's actually invested $100 million to set up a thing in North Carolina, and they're actually no longer in business. And part of that's because the FDA is not sure how to license this stuff that's continuous instead of yes or no answers. Okay. There's another aspect of this in cancer called methylation, so epigenetics. There are certainly some cancer types, colorectal cancer, for example, there are certain genes that become highly methylated, and there will be diagnostic tests based on that. Yet another area is forensics I haven't mentioned, okay? So not currently, but I do think that extra genomics will become a part of this medical care, not rapidly, but in a 10, 15, 20-year framework. You agree, David? Yeah. Thank you.